record the meeting and share my screen, and we're going to go through a PowerPoint. Van is stuck in a lab right now. He will be here soon, but I'm going to try to look at the chat and see if you guys have questions while I'm also teaching on the board. So gonna kind of be like a problem set thing. Oh, if you want to go, you can go. No, you're totally fine. All right, so yeah, if you guys have questions, you can drop them in the chat. Could somebody just give me a thumbs up or type yes in the chat just to make sure that you can all hear me? Okay, something's in the chat. Okay, awesome. Thank you, Tori. All right, so we are going to get started. This is like a topic 2 review, and then at the end, if we have extra time, we can take some questions. Oh. Thank you, Luke. All right. Okay, so I'm going to try to really project my voice so the computer can hear me also. Oh, and move this. Okay. All right, so we were going to start off with a little bit of a week 1 review. So I was going to have you guys kind of talk amongst yourselves, but you don't have to do that if you don't want to. You can just think on this. So we talked a little bit in class about beta barrels, and essentially they're just pore structures, cylindrical structures made of beta sheets. And a lot of times in the cell membrane, they're used as pores for ions and water. So because we talked about with the beta sheets, how you have your R groups alternating from side to side, what can you infer from that about the chemistry essentially of the R groups on either side of the beta barrel? And chat, if you have any answers, you can drop those too. What's your name?
Zach.
Zach? All right, what's up, Zach?
I think on the outside, for all the R groups that are facing out towards the environment, those would have to be the hydrophilic R groups.
Facing out this way or this way?
Is it going through the membrane?
It's going through the membrane.
So
this is embedded in your phospholipid bilayer as a pore.
Okay. So anything that I would say is exposed to the inside of the membrane, like the core of the membrane, would then have to be hydrophobic.
Right.
And anything not exposed to the core of the membrane, to the inside of the membrane, would then be hydrophilic.
Right. Okay. Yeah. So just to summarize that for anybody on Zoom that didn't hear, on the inside of your beta barrel, and hopefully you can see my mouse here, I'm not sure. On the inside, because you have polar water and ions going through, those are going to be charged or polar amino acids. And then on the outside, where it's embedded in the phospholipid bilayer, those are going to be nonpolar because they're interacting with your nonpolar lipid tails. And so essentially on either side of your beta sheets, you're going to have polar on one side and nonpolar on the other. Everybody good with that? All right. So, okay, protein misfolding. This is something that we talked about in class. And this is Van's slide. So, there are, I think, 4 or 5 things that we mentioned can kind of disrupt protein folding, make them fold incorrectly or make them misfold. So those were heat, salt, or just otherwise ion concentration, pH, detergents, and then any high energy wavelength like UV. And with all of those, for various reasons, they're going to basically disrupt the bond. So with your pH and your salt, those charges are going to maybe interrupt the ability of, say you have a positively charged R group and a negatively charged ion. Those may bind when they're not supposed to if your ion concentration is too high, just whatever, that could interrupt. And with heat, the kinetic energy, which we were just talking about before the review, could increase the energy in that system and essentially increase the movement. And shake them around so much so that the two things on the either side of your bond break apart. So think of it like if you're holding hands with a person, it's really easy if you're staying still or like kind of still, but once you start jumping up and down, you're probably going to drop each other's hands at some point. And that's kind of the same thing that's happening with the heat, how it increases the kinetic energy. So does anybody have like any questions on that? Also chat, and I hope you guys can hear me. No questions?
Like the pH is related to charge also?
Yeah, pH, because so yeah, like those are your acids and bases. So your acids are going to have like a negative charge, your bases are going to have a positive charge. Hi, 285. All right.
And detergents like
Yeah, detergents. So detergents, what they're going to do is essentially they're going to replace a bond. So like if you have germs on your hands, instead of having bacteria bond to your hand, and the bacteria is going to bond to the detergent. And you don't really have to know a lot of the specifics about why that happens, because there are lots of different kinds of detergents. So they like act differently in a chemical way. So you kind of just have to know that they're going to be like replacing bonds, disrupting existing bonds, and you know, creating a new bond with themselves. Zach.
When one polar thing can kind of steal a hydrogen bond from basically a hydrogen bond that already exists, is that because why is it able to steal the other hydrogen bonds? Like I read somewhere that it's competing effectively for that hydrogen bond, but I don't know really what that means.
Are you answering that? All right.
Would be like more like electronegative than what hydrogen's already like bonding with, because like hydrogen has like the partial positive. So like correct me if like I have like the periodic table wrong, but like if it was like already like bonding with nitrogen, then like oxygen may take its place because oxygen's more electronegative.
Right, yeah. So I don't know if Laura is going to talk about this, but essentially like you've already talked about like KD and stuff. So is your question like you have an existing bond and then something comes in and breaks that bond and replaces it?
Well, I guess
Is this
to
a
kind of
problem?
take an example. They mentioned urea,
Yeah.
how it disrupts the hydrogen bonds of water. But I suppose it's probably the oxygen.
Hi.
Hello.
Hi.
I hate physics.
I'm doing my best to balance the Zoom and this. So if you want to help me with either,
Yeah, of
those, I'll
course.
chat. All right. So sorry. Continue with your question.
But I was just confused how that works because in both cases, the hydrogen is forming a bond with the oxygen. So I was confused why the oxygen from like urea, for example, would take it from the H2O hydrogen bonds.
So I might have to look at that question a little closer after because I'm not sure which one is it. Two.
Just two.
Oh, okay. So we're. So I feel like that's a pretty specific problem set question. So I will come back to that at the end if we have time. And incentive for Zoom people to stay to the end and we can do problem set questions. Do you have another question?
Just sort of related. So like that doesn't like number two, like I guess like it wouldn't be like one of those like 5 examples like on the board that has to do with misfolding.
No, that's not misfolding. So I will come back to the problem set questions at the end. And then you can hit the next slide.
Yeah, of course.
All right. So chaperones, they're a type of protein. They're found in literally all organisms, all domains, like everything has them. I think even Laura said last semester all cells have chaperones, but that doesn't mean that every type of protein needs a chaperone to help it fold. So they're in all organisms, but not all proteins need a chaperone. And they have a couple of jobs. As proteins are being made and as they're coming out of the ribosome, they help those proteins fold. So essentially, like a lot of what they'll do, and I have a diagram for this on the next slide, is if you have a hydrophobic region emerging from your ribosome, it's gonna want to aggregate and essentially hide itself from the water-based solvent in the cell. So instead of letting it clump up before it's ready to fold, the chaperone's gonna come in, it's gonna bind to your hydrophobic region and protect it from the water in the cell until it's all ready to fold together. And then it'll release it and it can fold into its tertiary, maybe quaternary form. So then it's gonna, if you have a protein that's already misfolded or like unfolded, it can kind of do the same thing, can help stabilize those hydrophobic regions, it can protect it, it can provide a safe, stable environment for it to refold. And if it can't do that, if it's too damaged, if it's not efficient for the cell to expend the energy to refold it, then it can escort it to the proteasome. It's not doing the degrading, but it's taking the misfolded protein to the organelle so it can be degraded. And yeah, we talked about that. So they prevent aggregation, which is when essentially like you have a whole bunch of hydrophobic regions clumping together. So here is an image of that. So presumably these blue sections that our chaperone is bound to, those are all hydrophobic. And you have a whole bunch of chaperones. And then at the very end, once you have your mRNA fully translated, and your protein comes off, then the chaperones will unbind and you can fold into your tertiary structure. Does that make sense? Chaperone questions?
Do we need to know like how this works?
No, you just have to know that it does. So. All right, Dan.
All right, sorry about my delay. The physics lab ran late. So the heat shock response, there's two big responses we'll talk about to protein damage. Heat shock response occurs in the nucleus and the cytosol. Anywhere else is going to be another type that we'll talk about in the next slide. So as Lily just mentioned, proteasomes, they take these proteins and they degrade the damaged or unneeded proteins from the cell. Sorry, the zoom's in the way, but chaperones are also upregulated to assist in folding. This diagram represents it, I think, the best. So it starts as a nice oval, and then it has some spikes on it, and that's the aggregation. And then the purple little circles represent ubiquitin, which ubiquitin mediated proteolysis is just another way to degrade proteins. So it takes those edges off, and by the end, you don't have to worry about SUMO 23. It's just one of the many proteasomes in the cell. In the end, once all of those spikes are gone, it's in recovery and it's back to its nice oval shape. So that is the heat shock response. I think the most important thing to learn is that it occurs in the nucleus and the cytosol. Those are the big two.
All right. Okay, so the counterpart to heat shock response is UPR, unfolded protein response. We're not going to go into these too in depth because we're going to talk about these in class on Thursday. But so if your heat shock response happens in the cytosol nucleus, your UPR is going to be happening in the endoplasmic reticulum. And there are a couple of things that will happen during UPR. First of all, your misfolded, unfolded proteins are going to be retrotranslocated out of the ER and into the proteasome. So retrotranslocated just means that it kind of has to go through a backwards channel out of the ER, which is a little bit different to it. And then you're going to upregulate your chaperone production to kind of help either refold or escort those proteins back out so they can be degraded. And you're going to downregulate your translation because essentially like that will just decrease the load and the strain being put on the ER because you have fewer proteins coming through, so you can kind of focus on fixing the misfolded or unfolded ones. And then you're going to regulate your lipid biosynthesis, and that's going to increase your overall volume of your ER, so it can pretty much just like do all of these processes more efficiently.
Awesome. In protein ligand interactions, you probably have all heard of enzymes in Bio 151. That's this concept. So there's two big things that protein ligand interactions need. They need a specific amino acid properties. So arginine is a positively charged amino acid. So it cannot, if a hydrophobic amino acid came in and tried to bind, it wouldn't work. That's not a bond that would form. The other thing is the orientation. If we have a perfect shell, and a perfect ligand to come in, but if it's in the wrong orientation, it will not fit. So those are the two big things. It is very dependent on diffusion. If there's a high concentration of ligands in a cell, a lot of them will be bound to the proteins. But if there's a low concentration, it's much harder for it to diffuse in. It's not like a magnet where it'll, bring it in right where it wants it. It has to diffuse in on its own. So a higher concentration means more protein ligand interaction. And as shown in the picture, it doesn't have to be this perfect pocket. It could just be a flat surface and a protein and a ligand coming to bind on top of it. That's pretty good.
Okay, so here is kind of a problem set style question for you guys. This was on an exam a couple years back. So you can talk about this if you want, or you can just think on it. You can give us a thumbs up if you're ready to move on. But when you fry an egg, why is it that the whites turn from clear to opaque? And if you're totally lost, we have a hand. And we can also speculate. Go ahead.
Because it's getting hotter, so it's the proteins are unfolding and the hydrophobic parts are aggregating.
That was really good. Usually that takes
Brilliant.
Yeah,
Brilliant.
that was great. So chat, if you didn't hear, when you put an egg on a frying pan and you turn up the heat, you're increasing the kinetic energy, and that's going to cause, or you're increasing your heat. That's going to cause your bonds to break. You're going to expose your hydrophobic regions, because essentially your protein is like a ball of yarn that's being unwound, and all the knots in that ball of yarn, all the bonds are being broken. And then you have your hydrophobic regions that are going to want to clump together. And when they clump and they form aggregates, those essentially are what you're seeing when you're looking at egg white. They're so clumped together that they're, turning opaque. So that was.
So are covalent bonds breaking too?
No, your covalent bonds aren't breaking. So your covalent bonds are holding together your amino acids, and your non-covalent bonds are holding together your secondary, your tertiary, your quaternary structure. So if you, with the ball of yarn analogy, if you were breaking your covalent bonds, you would be ripping the yarn apart. But instead, we're just unwinding the ball of yarn. We're breaking the non-covalent bonds. So essentially you have a long string, and the hydrophobic regions of your string are all going to group together. Does that make sense?
Yeah, cool.
All right.
It's I saw a note from the other side, UV light is the only thing that really breaks covalent
High
bonds.
energy wavelengths can, if they're high enough, but
Yeah, and we'll learn about what happens when UV light hits it. It's pretty cool. No, not cool.
awesome.
All right.
Oh, huh.
All right. So this one was an exam question as well last fall. If glutamic acid in this diagram, so it's right here, if it were to be replaced by aspartic acid, which is this one, it's the same negative charge, but a little bit different shape and structure. Would the ligand, which is that it's cyclic AMP, still bind in the binding pocket? Does anyone have any ideas? It's a yes or no.
Thanks for coming.
Or maybe you can take there. I just had the wrong one.
Oh,
I want to get the B, because it's still this sorry, but I don't know if it has to be more specific than that.
That's a brilliant answer, and you are right. We would have to experimentally test it. Which would you err on, yes or no? Which do you think would be
I
more?
would err on yes.
And I that's the correct answer. That but you can't argue for both sides. What would be the argument that it wouldn't? Yeah.
It's not the exact right confirmation.
You got it. It's completely different size. That added carbon, it might not look a lot, but in the cell, everything is so specialized. Every binding pocket, every interaction is so specialized for one thing. And if adding a carbon, it might not look like it does much. It will impact it in some way. All right, I think there's one more as well. All right.
Oh, yeah.
Well, now what if it was replaced by arginine? Arginine?
I'd say arginine.
Does anyone have any ideas? Arginine is this one. So imagine this compound taking the spot of right here.
Is
What
that
is-
positive? Though?
That is a positive charge. Yep. So it's a positive charge, and glutamic acid is a negative one.
I would say no.
Yeah. Yeah. And why?
Because what's attracted to a negative charge is gonna be probably repelled by a positive.
You got it. And then I would also mention the structure. Because these two nitrogen groups here are super big and that adds it. It doesn't look it, but always mention the structure if you change amino acids.
Good.
Good to go. The next one.
Yeah, I'm just trying to transcribe a little bit. Alright, so we kind of touched on this before, but if you were to expose your protein to infrared wavelengths, would that disrupt either the structure or the function at any level? Why or why not?
Yes, okay, I'm seeing yes. Yes. Yes, okay. Anything in the chat? All right, no,
Actually, maybe not, because it's not as powerful even as visible light, it's not as high frequency.
I like your counter argument. So where we're gonna see disruption in the bonds is on this end of the spectrum. So these are all of your high energy wavelengths, and if you expose it to like UV or X-rays, gamma rays, any of that, then you're adding a ton of energy to the system. You're going to make your bonds unstable and eventually they're going to break. Kind of like what I was talking about earlier. If you're holding hands with somebody and walking, like you're not going to have a hard time not letting go, but as soon as you start jumping and adding that energy and moving around, then they're going to break. So we talked about this a little bit in class. Even if you didn't know that, you can kind of figure that out and extrapolate that, because this is your visible light in between your ultraviolet and your infrared. And we know if you expose a protein to visible light, you're not going to disrupt the bonds. Does that make sense? So anything below that is going to be too low energy to really have impact. Good.
Awesome. Last slide of the night. And go to problem sets. I think maybe I like. Affinity. So it's the strength of a protein ligand interactions. There's two big factors. We've already done some problems with them, but it's the number of interactions and the strength of the interactions, and also the structure of the interactions, which falls under strength. If a ligand forms 5 interactions with the binding site, it will have a higher affinity than one with 3 interactions. And then Laura likes to use this hypothesis called the Goldilocks hypothesis, where ligands need to be just right to fit into those binding pockets because they need every interaction to be super specific. So that's the big takeaway, is every interaction is so specialized that when you change one amino acid, it might not look like much, but it'll change everything. And all right, problem set time.
Just a small thing.
Yeah.
Can we say Goldilocks hypothesis on our problem set, or should we not?
No, I would say that ligand maybe you can.
If you say it, you would just have to explain.
If you explain it, say that this is the, has she gone over it in class?
Yeah.
Oh, then yeah, you should be golden. Just make sure to explain it because she likes, you know.
And also know, like, if you go to another class, Goldilocks hypothesis is not a thing. It's just a word that Laura uses to kind of describe that you want to be in that like intermediate zone. So. All right.
Perfect. All right, I pulled up the answer.
Oh, sick. Okay. And I told the chat that they can drop questions there too. So before
Lovely.
we get to problem set.
Yeah.
Yeah. Okay. Does anybody have any specific topic 1, topic 2 content related questions? Anything that you didn't understand from lecture or like our PowerPoint? We can do that first. We did
And
a
if you
fantastic
don't, and you
job.
if you have watched the pre-recorded lectures, the Thursday session will have all the information we didn't cover, like KD, and that's it. Maybe allosteric.
Yeah.
So that will be in the Thursday
The-
session.
Binding regulations.
Yeah.
Yeah.
Awesome.
What I'm hearing is we did a fantastic
Yes,
No.
you did a fantastic job.
job. No, I knew we did most of it. I did like figure this out.
Awesome. All right. Does anyone have any problem set questions, inquiries? Yes.
Like basically like 3 and
3 and 4. 3 has up to C. Do you have a problem with like A in particular or the whole thing?
for 3.
Yeah,
So I was saying ER, so now I feel more
Yeah.
confident about that after.
sorted.
this. What I was having trouble with was more like B and C. So I just said, I have so far they're escorted by chaperones, but do I need to go into a lot more detail?
You have like half of it. I don't want to say too much, but there's also a way that the cell can kind of flag a protein and give like a heads up to maybe the chaperones or whatever that it needs to go and be sent to the proteasome.
Is that ubiquitous?
That's it, yeah.
You got it.
And I think you also mentioned that like something about like energy like efficiency, like chaperones will escort the misfolding protein to the proteasome for degradation. If it's like whatnot like efficient for like the chaperones to like cover like all of like the hydrophobic mass.
Yeah, so like if you have, a couple proteins here and there that are misfolded or unfolded, it's not really a big deal for the cell to go in and try to fix those. But especially if they aren't a protein that's in high demand, or there are a lot of proteins that are being misfolded or unfolded or whatever, in most circumstances in the cell, you're going to just send it to be degraded. It's not worth the energy. It's not worth the risk of something aggregating when you have all of those unfolded proteins in the same place. So it kind of just depends on the circumstance. But as long as you know that there are multiple options for what could happen, then you're pretty much all set.
When does like each happen? Like, like when does like the cell pick like, oh, like the chaperone's gonna send it to the proteasome, or versus, oh, we're gonna tag it.
Again, I don't know if you have a better answer, but I think it's pretty much just like based on
dependent, yeah.
the circumstance. Like it's really case by case, because if you have, like I said, a ton of misfolded proteins, then the cell isn't going to want to individually unfold all of them. But if you have a really, really important protein that you need, that takes a long time to make, that takes a lot of energy to make, or there are like 1 or 2 that are misfolded, then it's going to be worth it for the cell to go and refold those correctly. So kind of just think about from an energy standpoint, what's most efficient for the cell, what's going to be the least risky for the cell, because it poses a lot of risk to the cell to leave a whole bunch of unfolded proteins in the same place, because then you can get your hydrophobic regions aggregating and that will kill the cell. So think about it like from the cell's perspective. What would it want to do to preserve energy and to protect itself?
For 3C, could I ask? So when it's talking about one cellular function that the cells want to decrease, and then one cellular function that it wants to upregulate. So what I had been thinking for this was decreasing maybe production of that protein. But I wasn't sure.
Yeah, so that's, I think, this slide, right?
Yeah.
Yeah. So not to like give you the answer or anything, but it's literally right up here. You're on the right track with down-regulating protein translation. It's not just going to be the one type of unfolded protein. It's going to be all proteins that you're down-regulating translation of, because that's going to decrease the load of proteins going through the ER. So everything is going to kind of be put on hold until they sort out, because when you have a whole bunch of stuff unfolding, that's like an emergency. The cell is going to want to allocate as many resources as possible to mitigating that as soon as possible. And that might just mean like down-regulating all translation.
Got it. All right, question in chat is, can you go back to the glutamic acid slide? Yeah.
I don't know why that did that, sorry.
What?
Why it animated like that.
Oh, yeah, no, not a problem. Luke, do you have any question or anything like that, or did you just want to look at the slide again?
Maybe repeat the answer.
You said give me a second, so yeah. It's also the one I had down in chat.
Oh, okay, yeah. Step ahead.
Yeah, that's it.
Also, we are very open to constructive criticism for you guys. And if you want to see something different on the PowerPoints, we're both doing this.
As you're probably shocked, we are both new.
like
Yeah.
as many pictures as possible.
Yeah.
Pictures is
Yeah.
really helpful.
The diagrams are definitely.
Yeah.
Have you guys used the movies section on Moodle? Yeah, that is for hard. I mean, we haven't done too much.
I
I
don't,
don't even
I don't
for
know, I haven't-
chaperones. Yeah.
I did not watch the movies from the movies folder until the
Wow.
first exam, and then I binge watched all of them. I really regretted not doing it soon. So if you haven't looked at them, especially if you're a visual learner, the movies folder is really helpful because I feel like sometimes it's hard to visualize stuff happening in the cell.
And the videos are like a minute, so
Right.
you you just get through all of them.
Right.
Fantastic. And you said you were also confused on 4?
Yeah, a few points on 4.
Any?
Oh, this is KD, yeah. All right, let's do it. Yeah, we didn't cover it. We know it, but it's Thursday's section might have a slide that will answer your question. I don't know if are you busy Thursdays on 7?
Not
Maybe.
that I know of.
Yeah, you never know.
Don't you want to come back to Moral at 7 p.m.?
I love moral. Yeah, that's where I have my best classes.
Oh yeah,
Most fun.
yeah,
Um,
yeah. What were you on, A or B or
um,
C, D or E? Oh my gosh. Okay.
yeah, it's a long. So, yeah, there were just like a few points on each one that like I was a little confused about. Like, I don't think I'm really answering it correctly for A. What in getting into the characteristics of the protein and its ligand that provides specificity. So I know, like we talked about, like it has to be able to make the proper interactions with the R groups of the ligand. But then getting into what dictates the KD of a protein, I started talking more about the portion of the lecture where she kind of got into much more specifics, like KD is given by this equation. This is the half-bound state, and what that means. And I don't know if I'm just going down a rabbit hole that's irrelevant and not answering the question.
So yeah, if you talked about the strength of bonds and what kinds of bonds, and that's what determines the KD. The half point is when half of it is bound. So they will go over that on Thursday, but that having a lower KD, do you remember what that means in terms of affinity? You got it. And so
So pretty much if you know affinity, you know KD. They work in reverse.
yeah, they're
So you don't
inversely.
really have to go down too much of a rabbit hole, just whatever you've already said. If you understand affinity, think about what the opposite of that would be for KD, you know?
So for like specificity of like binding, like you mean like go into the detail, like covalent bonds, like hydrogen bond and. like,
Yeah,
stuff
you
like
could,
that?
I would definitely mention
Yeah.
those and say like, you know, certain R groups will form some kind of bond. And then,
When you're talking about like especially stuff to do with amino acids, you don't have to come at it from like a chemistry class perspective, but mention the chemistry as much as possible. You want to be thinking about charge, you want to be thinking about bonding, you want to be thinking about sterics. If you're not bringing those things up specifically, you probably want to add some more detail to your answer, just as a rule of thumb, as an umbrella.
question in the chat. You haven't had time to watch the lecture videos yet. Explain the concept. Yeah, so in this question on the board, it's so a glutamic acid, as you can see in the bottom left-hand corner image, is a negatively charged amino acid, and you can see it on the right. I don't know if you can see my mouse, but it's hovering. It's that lighter green on the protein with the ligand interaction. And so if we're asking if the glutamic acid, the lighter green one, were to switch for an aspartic acid, which is also a negatively charged R group, would the ligand, which is the pink molecule, still bind in that binding pocket? And so you would have to test it to figure it out, but we can base it that it's the same charge and it's a very similar structure that we would argue that it would still bind to the protein. The ligand would still bind to the protein. But if you were to replace it with arginine, which is this yellow molecule on the top left-hand side of the diagram, if you were to replace the glutamic acid with an arginine, which is a positively charged amino acid, then we would argue that it would not bind because it wouldn't be able to form those interactions anymore. Because glutamic acid is negative and arginine is positive, and if it's forming a positive interaction already, positives will repel.
Any other questions from Zach?
Yeah, I just wanted to like go over the structure of urea.
Oh,
Is that 2?
Yeah.
Okay.
And so this is kind of what I was asking about from before. So I was assuming like the oxygen there has a negative dipole. And I was a little bit confused about what's going on the other end. Like, is there
With the nitrogens or the carbon?
the nitrogens bonded to the carbon? Like, is there a positive dipole down there because they're bonded to the? I know N and O kind of cancel each other out, but then the N's have those hydrogens.
I know, very fancy. All right, so you were saying that oxygen has a negative dipole. So the electrons would be going towards this way. And it would have a delta negative. What would make this?
Is that the nitrogen?
This is the carbon, sorry.
Oh, sorry.
Okay, that's the carbon.
So that would make the carbon partially positive.
You got it. All right, now let's look down at nitrogen. What's the electronegativity of nitrogen? Is it higher or lower than carbon? Is it more electronegative or less electronegative?
Higher.
Yeah, you got it. So take electrons that go this way. And so we would have a negative as well. And then the same over here. So that would make this guy super partially positive. But the nitrogens would still have a partial negative, even though, you know, this is super positive and it really wants electrons.
Okay.
Always base it on electronegativity.
And is that why it can compete for those hydrogen bonds that are happening between water? Like it.
The nitrogens or the,
It just says that like it disrupts the hydrogen bonded network of water molecules. It doesn't get in. I assumed it was the oxygen was grabbing some of the hydrogens from.
Yeah. Yep, the nitrogen can also do that.
Okay.
Yeah, because it has those lone pairs. It usually will be the oxygen because it has 2 lone pairs up there and this one only has 1. But yes, it will. I can't do that.
I would also recommend thinking about the sterics of urea and whether or not it can
Yeah.
easily fit into like a binding interface or a pocket. You know, whether that would make it easier or more difficult for people.
Yeah, definitely mention how big it is.
Always chemistry and sterics. You bring up one, you should bring up the other. Anybody want a picture of that before then?
Oh, please don't. This is no, go for it. No, no. A work of art.
You can come closer too if it's not.
I think I.
Chalk
Came out
one with
well
super
enough.
high contrast.
No, it is not.
I found that out earlier. I was trying to like write a little heading. Very
Yeah.
Writing in chalk.
So basically just to like confirm like high concentrations of urea can like unfold proteins because of like the dipoles. So like so since like nitrogen is like partially negative, like it would be like more able to like grab hydrogens.
Yeah, it would. In this one would be probably the main antagonist of the story. It's really electronegative and it loves electrons. And so let's say it finds. No, it's not beginning. But let's say a hydrogen's over here, and it comes and it takes that hydrogen. It can do that pretty easily because it's so small, and it can go in and out and grab those hydrogen molecules.
Like oxygen is small, hydrogen is small.
This, like in terms of a whole alpha helix with, you know, hundreds of amino acids, this tiny little urea, if it was grown to, if it was to size, it would be like that. And so it can go in and out just really easily because it's so small, and that makes it really versatile.
Oh, okay, that's cool.
This is kind of getting back more at, when you were talking about the, unfolded protein response. So one thing that you talked about was upregulation of lipid biosynthesis to increase PR and membrane size. And I'm just wondering why, like, what is it?
So we don't go into a ton of detail on this. And I had to look this up because I didn't have that on my notes from last semester. We literally didn't talk about it. So I don't think that you need to know much beyond that it does that. But part of it, I believe, is to make more volume in the ER to accommodate the fact that you're gonna have an influx of chaperones. And Ben, do you know anything else about this other than that? Like, I
No,
feel like
we
don't
didn't go
really
much
talk
into
about that.
depth. Yeah, no.
And I did, like, I was really getting off into the weeds last night, like, I was looking at, like, journal articles, whatever, but there's not a lot of information that I can readily find at the level of this class, so. But feel free, if you're not satisfied with that response, because nor am I, to ask Laura about that. I'm probably gonna do the same thing. So I'm excited to.
Yes, have you guys been to our small classroom hours on Fridays at 2 p.m.?
Um,
You have classes.
yeah, I have like classes during all the,
Oh.
sessions.
Oh,
It
no.
sucks. That's why I'm bombarding you guys.
No, you're good.
No, yeah.
If you don't wanna go, or if you can't go to Laura's, we also have, all weekday reviews, so I don't know if you're booked for all of those, but
Yeah, my schedule this year sucks.
wow.
Oh, no, wow.
Yeah.
Alright, also feel free to always send me notes. I think probably most of the TAs will be willing to answer problem set questions at the end, but once we get into more content heavy topics, there will not be as much time at the end, so always feel free to like shoot us. a note. Anything else? Feel good? Is this worse or better than topic one?
I personally thought it was like a little more difficult topic one, just because topic one I went over a lot last year.
Yeah.
This is true, yeah. For whatever reason, in our class last spring, like a lot of people had not covered the topic one stuff at all when we did it in a week. So the topic one stuff on our first exam was not
That's
good.
true. You guys are in a very good spot. You've gotten some problem set question answered. It's only Tuesday. We've got
Right, yeah.
the rest of the week. And yeah,
Right, I think we'll probably wrap up. If you have any questions after, and I know you mentioned research, so if you have any questions about that, I don't know if I'll be able to help you, but always, feel free, we can stay and chat or whatever.
Yeah.
And chat, if you guys don't have any more questions, I'm gonna stop the recording and I think end the meeting. So I'll give it like a minute. Okay, yeah, thank you guys so much for coming. We
Yeah,
have
thank
another
you.
one on Thursday, 2 different TAs, but you should still come because we'll probably have some more content further in the week. All right, bye.
Thank you.
Yeah, thank you. Okay,
Fantastic.
now Van.
how did you set this